Folks. Welcome to lecture 6. Open up the PowerPoint and share my screen. All right, we're going to start talking about correlations and regression. So far we've covered the one sample t-test, the two sample independent t-test, as well as the paired t-test or matched t-test. And we went over ANOVA and post hoc tests in lecture 5. So now we're going to do correlation and regression. So this is when we compare two continuous variables. So that means they're continuing to count or continuing to move along. It's a continuous measurement possibly. An example down here is seeds and mass. So as the number of seeds go up, the amount of mass goes up. This is the correlation line here. And the distance between these dots and the line has to do with the correlation coefficient, which we are going to talk about. The difference between a correlation and regression, not aggression, is whether or not it's a causal relationship. So do you expect changes in variable X to cause changes in variable Y? So do you expect the number of seeds to cause the mass of the seeds to go up or down? If that's true, then it's a regression. If it's not, then it's a correlation. So for an example, does being taller mean you're going to have higher test scores? No, it's not a causal relationship. It could, show up that that's the case, but we can't say that that is a causal relationship. However, we know that if you eat a whole bunch of donuts, you're probably going to gain weight. So that is a causal relationship. If you don't expect it, it's called a correlation. Okay, it just happens to be that 90% of the population at CSUCI drives a white car. It doesn't mean that going to CSUCI means that it will cause you to drive a white car. Does that make sense? Okay, so if it is a causal relationship, we do expect that donuts cause weight gain. We're going to run a regression analysis. So when you first get your data, you're going to make some graphs, right? We've learned how to do that. And you go ahead and look at these graphs here, and A and B are positive relationships. C is there's no relationship at all. And D and E are negative relationships. All right. Correlation coefficients is basically how close to a perfect line your data is and the direction of the relationship. So there are parametric and non-parametric correlation tests. We will go over this in detail. All right, so correlation coefficients, again, we have our null hypothesis, meaning that there's no relationship. And our range is from -1 to 1. You'll see that better in this graph here. So if we consider that these are all graphs, 1, 0.8, 0.4, 0, -0.4, -0.8, and 1. So this is the range of, or the strength rather, of relationships. And then the direction is either from left to top right. Those are positive. 0 is a neutral, and then these over here on the right are negative when it starts from the top left and goes down to the right. Those are negative relationships. So this is the math behind the Pearson's correlation coefficient rather. You do not have to memorize how to do the math, but you should know what it means and what is behind it. You do have to make sure that your variables are normally distributed before you run this test. Just like every other test, you always have to make sure your variables are normally distributed. So we're going to look and see if height and self-esteem are correlated or not. We go through the math, take the numbers, and we end up with an R value of 0.73. All right. So when we graph that, it looks like this. As self-esteem goes up, height is going up as well. So that's a pretty decent R value there. So is it a significant correlation? And this is when we figure out if we're calculating R or P. I'm going to share this paper by Ambrose that talks about whether or not the value is different enough from 0 and how you decide. Sample size and correlation is a big deal, right? So we've talked about sample size a little bit and some other things. You need a pretty decent size sample. Otherwise, you are going to probably reject your null hypothesis, right? If you only have 5 samples, it's not going to be able to show you much, right? So less than 10 is not awesome. You need more than 10 samples in order to even run a test like this. All right, our correlation coefficients. Well, I'm having a hard time talking today. In R, we run the COR function to calculate these variables. I've put all of these codes in here for you. This is the code that shows you how to do the correlation coefficient. This is between hours and score. I'm going to share some data with you, and we will do a tutorial together, and we just have all of this in here. This is the Pearson correlation coefficient between hours and score. That's 0.86. So that's pretty decent. Listen, if you have NA values in your data, you can use the argument equals complete observation to only use the rows where there are no NA values. That's just something to put in your CAP for later. We can calculate our correlation coefficient to ignore NA values with this code down here. Okay, so this is basically running a correlation between data frame X and data frame Y. I'm going to give you that data frame, so just relax for right now, and I'm going to show you this again later. This is how we create our data frame. We have practice exams and score, then we look at our data frame, then we're going to go ahead and do the Pearson correlation coefficient. All right, we're assuming that our data is normally distributed. It is because I'm giving it to you. Once we calculate it, you go ahead and run the data. This is the output that you have. You have hours, practice exams, and scores. This is how we read that output. The correlation coefficient between hours and practice exams is a negative 0.13. Between hours and scores is 0.86. That's pretty high. Correlation between practice exams and scores is a negative 0.39. That's a bummer. So if you're taking practice exams, your scores go down. So the correlation coefficient between each variable always is 1, and we just look at the value along that diagonal to see how close they are, and that's how we figure out the matrix. So calculating the Spearman correlation coefficient is when, right, Spearman is different than, okay, I'm going to reiterate this. I will go back because I can tell you can't remember, and that's okay. So there are 2 correlation tests, right? So here's Pearson's, which is the main one, and Spearman is non-parametric. Okay, that means the data is not normally distributed. Got it? All right. I just want to make sure we're all on the same page and you don't forget. So we have Pearson's and Spearman's. So Pearson's is for normally distributed data. Spearman's is when you do not have normally distributed data. So it's the same concept, correlation, data frame X and Y, but our method is going to be Spearman because the default is Pearson's. So you have to tell R to use Spearman. Okay. Again, our DF is, data frame is hours and practice exams. Our Spearman correlation coefficient turns out to be 0.125. All right. We can also use Kendall's correlation coefficient between values. And this is when you want to use the Spearman correlation because your data isn't normally distributed, but your sample size is really small and there's many tied ranks. And that's something that you're going to learn more about in a minute. So just take a breath. I know this is a lot, but just kind of put these things in your hat for later. Okay, there is another option for correlation coefficients if you have a small sample size. So again, correlation versus regression. If you're eating donuts, you're gonna gain weight, right? That is a causal relationship. So you cause a relationship, that's a regression. A linear regression is the math, okay? So what we do is you look at the causal relationship between 2 variables, right? So we have X on the bottom and Y on the left. And the question is, can you predict the values of Y based on X? Maybe, and that's why we run a linear regression, all right? So you might remember this from algebra is your slope equation, okay? Y equals A plus BX. BX is our slope. A is our y-intercept here. Y is always the dependent variable, so it depends on X, and X is always your independent variable. This is something that you need to know. You need to always make sure that you graph properly. So write this down, snapshot this slide, whatever it is that you need to do. But X is always our independent variable, okay? And Y is your dependent variable, all right? X, independent. You're like, well, what the heck does that mean? All right, we'll go over that in just a second, okay? Depends on X. See, I wrote it down too. Write it down. Linear regression is this model. So y sub 1 basically is this here, a plus bx plus e. Okay, so e is the error. So that's the difference here, right here, these little blue lines. The difference from the line to a typical data point. There is a way to calculate that as well, which is called the residual, which we talked a little bit about in the previous lecture, but it's not explained by the specific equation. What you have to go over is what's called a best fit line, all right? So we drop E from the general equation. We don't worry about that too much, and we just use y equals a plus xb and run the regression line, okay? Once you get a regression done, I will show you in R how to calculate the residuals. Actually, I think I showed you how to do this already in the post hoc test for ANOVA. It's very similar for a regression. You run a post hoc test, R measures the distance from all the line and creates an error for you. In the linear regression here, you can see as x is on the bottom, y is on the left, here is our line, and this red line, this dotted red line, is where x has no effect on y, and y does not depend on x. But once it gets above this line, so it looks like 4, right? So we get out to about, once it changes over, so when x gets up to 2 and some change, that's when it starts having an effect on y. Does that make sense? So our null hypothesis is that b is 0. So that's this effect on x, all right? I know it's a lot of math. It's a little weird yet, but just give me a second. I'm going to give you some examples that make more sense. But you have to learn the math. So a statistical analysis, we can use the ANOVA model. We did this, right? We did an ANOVA in the last lecture. B is the slope of our regression line. This is the slope. We get this distance. This gives us the error value. So say you want to show the error or the residual or error bars. This is how you figure that out. So F is our variance. So do you remember what variance is? So it's variance of the regression line or variance due to error. There are two kinds of variance in a linear regression. So degrees of freedom go in the numerator, okay, is 1, and degrees of freedom in the denominator is always n minus 2. So depending on the math, you just have to write these things down. R is going to do all of this for you. You do not have to do any of this by hand anymore because it is not 1980. It's kind of awesome. So even though R might be hard and a pain in the brouhaha, you do not have to do the math by hand. Now, if you're like a math person and you like it, awesome. I never have been. I have had to learn and it's not easy for me. So I really appreciate programs like R. Okay. You just need to understand the concept that the direction of the line is the slope of the line, like a ski slope or the slope of your driveway, okay? The slope, that's what that is. A linear regression is a statistical test, okay? When you get a p less than 0.05, that means that the slope is not 0, right? It's not straight across. It means that it goes up or down. So that means that there is a difference. So instead of a specific difference between like group 1 and group 2, you're trying to see if group 1 changes group 2. Does that make more sense? So do the number of flowers on the purple sage bush increase or decrease the number of bees that come to, or does it change the number of bees that come to the bush? Okay. So we're not comparing like how many bees and how many flowers per se. We're trying to see if one causes the other, and that would be a linear regression. So your null hypothesis is that it doesn't matter. And your alternative hypothesis is that it does. So if you run your regression and your p-value is less than 0.05, that means that there is a difference and y does depend on x. So the number of bees depend on the number of flowers or vice versa. We'll have to talk about that. It's actually in your review questions, there is a problem like that. So I want you to pay close attention. So the R squared coefficient, you may have heard of this before. So this is how well the regression equation explains the variance. So the variance, again, is the variability of the points around the line. So 0 is they're all over the place. 0.5 is they're kind of close. And 1 is they're exactly on the line, okay? We interpret it as a percent. 1 is 100 of the variance, you know, that all the points fall on the line. It's about 50% and 0%. So your R squared value, the higher it is, the more definitive you can answer the question of X causes Y, okay? Or X causes changes in Y. So when we're reporting the results, you need to give an F value. to make sure you put that out, you have a p-value, you have a y-value, and an r-squared value. There's no significant function in a relationship between y and x based on these numbers, okay? So our p-value is 0.9, so it's above 0.05, so that means there's no significant relationship between those two. All right, you can also write variable y does not depend on variable x based on these. The linear regression equation is y equals these numbers here, and it really only explains 0.1% of the variance in the data because our r-squared value is 0.001. So that's really, really small. Does that make sense? Hopefully it does. This is what you're going to need to make sure you do when you are reporting a linear regression. The assumptions is that there is a relationship between A and Y, that the observations are completely independent. So the pairs should be from a different object, unlike in a t-test where it's paired and we're measuring the same object before and after something. Can't be like that. A linear regression has to be completely separate objects. Homoscedasticity is constant variance, so it means that your residuals should not be correlated with the change in x. All right, so it means that the distance from that line should not correlate with the samples on your x line. Okay, and we'll talk about how to check for that as well. The data needs to be normally distributed, and the residuals should also be normally distributed. Y should be normally distributed. So these are all things that you're going to learn how to test for. There are a lot of assumptions. If you run a linear regression and you do not check all of these things, it doesn't count. Okay, it's bad math. And a lot of scientists do this because they don't know better. If you down the road, after you take this class and you're doing a project and you can't remember all of this stuff, you should download all these PDFs so you have all this in your files somewhere. But you can always reach out and ask me, ask another professor. Don't just run your stats without asking for help. When I was in grad school, one of the professors on my committee, that's all he did was stats. And so whenever I ran into an issue where my colleagues and I, my other grad students, we could figure it out. And even today, same thing. We go to somebody who really knows what they're doing. Like, I've gotten better at this, but I always have friends that they're so smart and so good at this. And, I help them with certain things. They help me with certain things. Do not try to do this completely on your own. For this class, it's different. You're learning. I'm trying to give you practice exercises to go through this, but write these notes down or download the PDFs and save them somewhere. All right. There's my soapbox about stats. Okay. So in a linear regression, if you don't have normally distributed data, you can transform your Y variable. So if you have some outliers, you could remove them, but you need to be careful doing that because sometimes outliers can cause problems, but sometimes it's a big part of the data. So say you have, like here in graph A, all your samples hit this line except for this one rando over here, but your line is still has pretty good, probably a pretty good R-squared number, so you wouldn't take that out. However, if this dot, like in B, skews your line, then you could take it out. It just depends though, because you have to explain why you took that data point out. We always want to have complete answers. This is the example of a complete answer. To meet the assumptions of normality in variable y, it was log transformed. We'll talk about transformation of data in another lecture because it was removed, blah, blah. This is what you would write out, okay? Linear regression. It's a technique that we can understand the relationship between a single explanatory variable and a single response variable, okay? So it finds the line that best fits the data. For the first part in R, you will load the data. So we're going to create a fake data set, okay? Following two variables for 15 students, total hour study for the exam and the exam score. Okay, we're going to attempt to fit a linear regression model and see if the hours are an explanatory value of the score, right? So the more hours they study, do their exam scores go up or not? Okay. So the following code, here we go. You can copy and paste this into R. You click on and copy and paste here, you'll get your first 6 rows of data set. You're going to want to attach your data frame. So make sure you run all 3 lines of this code. So we want to see if the relationship is linear. So first we're going to create a scatter plot. So you want to visualize the data. So I want you to go ahead and copy and paste this code in and run the code, and this should pop out with a graph that looks similar to this here. Okay. And we can see that the relationship's not totally linear. Let's try a box plot and see if there's any funky outliers. So, oops, I forgot to put this in blue. Here's the code right here. Oops, go back, Dorothy. Here's the code, box plot, and you're gonna do score. So go ahead and copy box plot score. Don't copy the dot or it'll mess up an R, and you should get this box plot. We don't see any outliers, so there's no tiny circles like outside the box, or excuse me, which means, or inside the box or any kind of weird thing. So there's no outliers in our data set, so that's not an issue. Now we're gonna go ahead and do the linear regression. So on the left-hand side of the screen, it says fit simple linear regression model. Go ahead and copy all 3 lines of this code. The model is the LM. That's what linear regression model is. Then you're comparing the score to hours. You're going to view the model summary. So you have to summarize the model and then go ahead and run this formula here. So there's no outliers, so we can do a regression analysis. We're using hours as our explanatory variable. So hours is our Y, okay, Y variable. And score as the response, excuse me, hours is our X variable rather, and sorry, score is our Y variable, okay? So does the amount of hours on X affect Y? All right, let's go ahead and run it, and then this should be your output here. So we have the quartiles here, the coefficients, standard error, our significant codes, multiple R squared, adjusted R squared, the F statistic, degrees of freedom, and our p-value. All right, so what does all that mean? So from the model summary, you can see that the score equals 65.53, which is right here, okay, plus 1.982 hours, all right? So this means that for every additional hour studied, the average increase in exam score is 1.9 points, okay? And the intercept value is that spot where it meets on the graph where the changes start to happen. So at the intercept value of 65.3, it tells us the average, that's like the average score if they don't study at all. So they'll get like a 65, which is a D. Okay, that makes sense. So we can also use it to figure out what an expected exam score is. So for example, a student who studies 10 hours is expected to receive about an 85. So you take that intercept value plus our 1.92, which is that average increase, which is you get here from the hours estimated standard error here, okay? Then times 10, which is the 10 hours studying, and that's the 85.5. So it's kind of cool how you can see that. So our p-value is way below 0.05, so it's definitely affecting it, right? The hours that they study definitely affects the exam score. So here's how we interpret all of that. P-value is associated with the model coefficients. Our multiple R squared tells us that the percentage of the variation of exam scores can be explained by the hour study. Our residual standard error is the average distance observed from the regression line. And then our F statistic and p-value, okay, is the overall significance of the regression model. All right, so now we want to create a residual plot. All right, so the last step is to look at this because we've done the linear regression, but there's all those distances, right? And I talked to you that we need to figure out if the regressions are normally distributed and homozygotic to each level. If you want to find out more about that, I have created these links in the slides. So to verify that everything's been met, you want a residuals versus fitted values plot. So this plot is really good for confirming homozygosity. The x-axis down here displays the fitted values, and the y-axis displays our residual values. As long as the residuals appear to be randomly and evenly distributed throughout the chart, like the line, see, goes pretty much straight across or even just slightly up or down, then that means that we can assume that homozygosity has not been violated. If this middle line here looks like your other lines, like it has a big angle one way or the other, that means that you do not have homozygosity and you cannot use your residuals. You cannot, your plot, something's wrong with your data. All right, so in order to run this, you go ahead and copy this code here on the left. This will give you the residual model. This will fit your residual, so it'll give you a plot over on the side. your plot will pop up, and then when you run the AB line, it will give you this middle line here. So our residuals appear to be randomly scattered around 0, which is great, and they don't exhibit any normal patterns. So then we can also make what's called a QQ plot for residuals. So we want them to fall around a roughly straight line at a 45-degree angle, and that way we know the data is also normally distributed. So go ahead and copy and paste these QQ plot residuals, all right, these 2 lines of code. You should end up with this normal QQ plot on the right. All right, and so you can see the residuals stray, like they're kind of like dotted all over from the 45-degree line, but not enough to cause serious concern. If you had them kind of all over the place, that's when we know that would be an issue, and the residuals are not normally distributed, and then we couldn't use our model basically. So this is the complete R code for our linear regression that we did today. This is the data set at the top. You can copy and paste all of this right into R, and it will give you all of the things that I just talked to you about. And we're all done.